Dear students, welcome to Bluepin Online. Shall we begin? Please use headset, pen and paper to take notes.
Here you go. Please let's know your feedback after class.
We can entering into DNA repair. Okay, damage to DNA that is caused by replication errors or mutations have serious consequences. The cell possesses an inbuilt system to repair the damaged DNA. It is known as DNA repair mechanism. Okay, Sandhya?
Yes.
Okay.
Yes.
There is proofreading mechanism, but there is a chance for replication error or mutation.
Yes, ma'am.
That will cause serious consequences. So,
Yes, ma'am.
to avoid that consequences, the cell possesses an inbuilt system to repair the damaged DNA, and it is known as DNA repair mechanism.
Yes, ma'am.
Fidelity of replication is very high, but there is a chance for error. One error per billion base pairs during each cycle of replication. That much important process. So that's why one error per billion base pairs.
Yes.
Okay. Besides possible errors in replication, there is a chance for one error per billion base pairs. Besides that, the DNA is constantly attacked by physical and chemical agents.
Yes. ma'am.
That includes radiation at the time of when we exposed to radiation. Then when we exposed to free radicals are formed within our body. So free radicals and also chemicals. Okay. These are the agents of chemical and physical agents that causes DNA repair. If it is not repaired, permanent mutation will occur that will lead to cancer. So how cancer forms? We know it's a genetic reason is there. So many reasons are found out for cancer. One of the reason is this, because of if it is not repaired, this DNA replication, what happens? Permanent mutation will occur that leads to cancer.
Cancer.
Okay, that's about the introduction of DNA repair. Now we enter into different types of DNA repair that occurs in our body. Okay.
Yes. ma'am.
So, through which mechanism this DNA repair occurs? Okay.
Yes. ma'am.
There are four mechanisms how this DNA repair occurs. Okay. First of all, we recognize the distorted region of DNA. Okay. We
Yes.
discover the distorted region of DNA. Then removal or excision of the damaged region of DNA strand. Simple process. Okay.
Yes.
Then filling of gap left by the excision of damaged DNA strand. Sealing of nick in the strand that has undergone repair by ligase. Okay. Almost it is very simple process, but we explain in 4 sentences. First of all, we find out the error, then we remove that error, then gap filling occurs. There is a nick, and the nick sealing is done by ligase. Simple
Yes.
process.
Yes.
Okay. But
Yes.
the molecules involved in each and every repair is different. Based on the difference, we can classify DNA repair into different types. The basic mechanism is the same. Okay.
Yes.
Okay.
Yes.
Okay. Then next, there are different types of repair system. Have you heard about this name of this repair system, Sandhya? Plus
No.
2 classes, or in some cases, we can see this name. Yes.
Mismatch repair.
Yeah, mismatch repair. It's very common repair. Mismatch repair, base excision repair. Okay. Then nucleotide excision repair. Then direct repair. Then next one is homologous recombination and non-homologous end joining repair. Okay.
Yes. ma'am.
Simple to complex. Okay. Like that. Mismatch repair. If there is mismatches, that coping error. Base excision repair, there is a chance for abnormal bases. Which are the normal bases in DNA?
AGTC.
AGTC, adenine, guanine, thymine, cytosine. Okay.
Yeah.
Instead of adenine, guanine, thymine, and cytosine, there is a chance for the existence of abnormal bases like uracil, hypoxanthine, xanthine. Have you heard about hypoxanthine and xanthine?
Yes, ma'am. That
In which topic? In which topic?
nucleotide synthesis.
Yeah, nucleotide synthesis. In which metabolism? Purine or pyrimidine?
AG, that purine.
Purine. What is the end product of purine metabolism?
Uric acid.
Yes. Accumulation of uric acid that leads to one disorder.
Gout.
Okay. So there is a chance for the existence of abnormal bases like uracil, hypoxanthine, and xanthine in case of base accession repair. So we have to remove that abnormal bases. Okay. In some cases, that's about abnormal bases. In some cases, there is a chance for the formation of pyrimidine dimers. Which are pyrimidines? Which are pyrimidines present in DNA?
Cytosine and thymine.
Okay, the cytosine and thymine dimers can be there, and some cases alkylation occurs. Okay, so this abnormalities should be removed. Otherwise, that will lead to mutation and also to cancer. Okay, so when we remove this type of abnormalities, it is known as base accession repair. It's a particular mechanism, same step, but the molecules involved in that repair is different. Base accession repair. The third one, it is known as nucleotide accession repair. Okay? Yes. DNA lesions that cause large structural changes. If there is any kind of DNA lesion occurs that causes large structural changes in DNA. Okay?
Yes, ma'am.
Okay. Pyrimidine dimers that may be due to chemical radiation, chemical agents, radiation, or spontaneous. Pyrimidine dimers are formed, and that pyrimidine dimers causes DNA lesions, and that cause large structural change. Okay? Yes. Then the fourth one is DR, direct repair. In case of direct repair, the type of damage we can see, there is a chance for pyrimidine dimers formation, O6-methyl guanine formation, but it's a simply repair, direct repair. Okay, the DNA itself contains no more mechanism, it itself changes that repair. That is known as direct repair.
Yes.
Okay. Then next, what is meant by recombination of DNA? We are not entering into that topic. What is your idea about the recombination of DNA?
That homologous chromosomes, one part of them, they exchange the genetic.
Okay, so that we will discuss in detail. Now, just understand homologous recombination and non-homologous end joining repair. In that cases, we need double strand breaks to repair. Type of damage, what happens? Double strand break occurs. Okay?
Okay, yes.
Okay, Sandhya. So it's an overview. We discuss one by one. Next, so mismatch repair, mismatches, scoping error, base accession repair, abnormal bases, uracil, hypoxanthine and xanthine, pyrimidine dimers and alkylated bases, nucleotide accession repair, DNA lesions that cause large structural changes like pyrimidine dimers formed due to chemical radiation or spontaneous, direct repair, two factors which are the pyrimidine dimers and O6-methyl guanine. Then the last type of repair, that is homologous recombination, HR, and non-homologous end joining repair. In these cases, DNA double strand breaks. Okay.
Yes.
Then now it is the interaction time. Any doubt? Any doubt about this topic? You understand what is DNA repair, what are the mechanism, and which are the types. Okay. Then explain definition of DNA repair.
DNA repair, it should occur in a high fidelity process. It has to change the damaged part of the. It's an inbuilt cell process system. There are chance of errors in errors per billion base pairs.
One error per billion base pairs.
One error
Okay.
per billion base pairs.
There is also a chance for chemical and physical.
Physical, chemical, and radical, that free radical constantly, the DNA is constantly exposed to that. So if there is any error and not repaired, there is going to be permanent damage, that is mutation, which leads to cancer.
Cancer. Okay. Which are the 4 mechanisms of DNA repair? 4 steps.
The first, it has to recognize the DNA, the region of DNA. Then there should
Which
be
region? Distorted region of
distorted
DNA.
region of DNA, and then there is going to be removal of excision of the damaged part of the DNA.
Okay.
Then there is filling of gaps by the of the damaged DNA. Then
By
there
which
is
enzyme? Filling the gap by which enzyme? Normally, in case of replication, which enzyme? Polymerase. Okay, DNA polymerase enzyme, generally filling of gaps. Then what about Nick? Nick joining?
Ligase enzyme, which joins the nick.
Okay. Then Sandhya, just name the types of DNA repair. Just name. No need to explain more. Just name. We are not entering into detail. That's why just mention the names of types of DNA repair.
Mismatch repair, base excision repair, nucleotide excision repair, and direct.
One more repair is the
homologous recombination and
homologous recombination and non-homologous end-joining repair. Okay. Okay. Then next we are entering into the first repair, that is known as mismatch repair. Okay. Mismatch repair. What happens is that first we are going to study the steps, then we can see the diagram. Strand directed mismatch repair occurs. That means there are 2 strands in DNA. Yes?
Yes, ma'am.
One parent strand and one newly synthesized strand. Okay?
Yes, ma'am.
Yes. There are some specific proteins that is involved in mismatch repair. I already told you that proteins or protein factors are different in all kinds of repair. The mechanism is almost the same. So here, in case of mismatch repair, specific protein that scan the newly synthesized strand. Okay?
Yes,
so how can this protein identify this newly synthesized strand? There are 2 strands, newly synthesized strand and parent strand. So this protein scan the newly synthesized strand. Why? Because new strand is identified by not being methylated. Do you remember? In our eukaryotic replication, one step is there.
Yes, ma'am.
Yes?
Yes, ma'am.
Yes? I already told you, and that time I told you there is a importance for this methylation. Yes?
Yes,
so parent DNA strand is methylated, but newly synthesized strand is not methylated. Okay?
Yes.
Which enzyme is used for methylation?
DAM methylase.
Okay. We are not entering into that much. It's a deep area. When we study molecular biology in detail, there is a huge portion for the action of DAM methylase. Now you just know there is methylation occurs with the help of one enzyme, DAM methylase. Parent strand methylated, newly synthesized strand is non-methylated. So the specific protein that is involved in mismatch repair that scan the newly synthesized strand. Okay?
Yes, ma'am.
Okay. So mismatched area is identified. A loop is made. Okay.
Yes. Yes, ma'am.
In E. coli, in prokaryotes and also in eukaryotes, there is recognition and looping is done by 3 proteins, mute S and mute L. It is mute C. It is not mute C. It is mute L and mute H. Okay. When this protein binds, that segment is removed.
Yes, ma'am.
Okay. After the binding of protein, that particular segment is removed. Finally, the correct segment is synthesized with the help of the last 2 steps of our mechanism of repair. Finally, correct segment is synthesized with the help of DNA polymerase, SSB and ligase. Yes?
Yes.
Mechanism is clear. Then, which is the disorder that is associated with this mismatch repair? One disorder we discuss associated with telomerase replication, which is that?
Progeria.
Okay, like that, mismatch repair. The disorder associated with mismatch repair is hereditary non-polyposis colon cancer, or it is known as Lynch syndrome. Okay,
Yes.
one of the most common inherited cancer that is linked with faulty mismatch repair. Okay, yes. Then next, we can see the diagram here. Okay.
Yes, ma'am. Yes.
This is the parent strand and daughter strand. So what happens here? A loop is formed, error in newly made strand, binding of mismatch proofreading proteins, green, blue, white. 3 proteins are bind. One loop is made. Okay.
Yes.
From the diagram it is clear. The loop is made. Then this strand is removed. You can see the strand is removed and corrected strand occurs. Okay.
Yes.
So this mismatch repair is also known as strand directed mismatch repair. Okay. We direct the strand. So direct the strand with the help of methylation, DAM methylation, by the action of DAM methylase. Okay. So, okay. Once again, what happens? Error in newly made strand, binding of mismatch proofreading proteins. Which are the proteins? Can you name it?
Ma'am?
Can you name it?
Proteins which are involved.
In this picture, it is clear.
And,
yes?
Yes.
In this diagram, it is more clear. You can see this parent template strand, its upper strand. The difference is there. Normally, we draw the diagram of DNA, parent strand, lower and newly synthesized strand upper. So, here, parent template strand. Okay. Then, what happens? Recognition of mismatch by mute S, mute L, and mute. It is mute L, not mute C. Okay. Once again, I repeat, mute L, mute H. Then what happens here? The cleavage of backbone in the vicinity of mismatch and removal of P by exonuclease 1. Okay. Synthesis of new nucleotide by DNA polymerase 3. Clear?
Yes, ma'am.
Yes. Okay, that's about our mismatch repair. Any problem with mismatch repair? We can see this methylation. Yes, CH3 is attached. Can you see this?
Yes, ma'am.
CTAG 5' end, GATC, cards, GATC. Then this adenine is methylated. Adenine is methylated, and this methylation is done by DAM methylase.
Yes.
That's why this scanning occurs. Parent strand is scanned by the presence of this methyl group. Okay? Yes. Then next is the question time. Are you ready? Yes. Sandhya. Then, what is the role of DAM methylase?
The DAM methylase that forms the methyl in the newly synthesized strand.
And methyl group. Methylation occurs in parent strand or newly synthesized strand?
Parent strand.
Okay. Look at the picture. You can see parent template strand.
Methylation.
Any confusion? Any confusion, Sandhya?
No.
Simple mistake. Okay. Parent strand is green in color and newly synthesized strand is red in color. Okay. When we discuss eukaryotic replication, at the time of initiation, we contact, when we find out origin or IC, we contact this methylation. Okay.
Yes.
That methylation occurs in parent strand. After initiation, elongation, and termination, we synthesize new strand. Okay.
Yes.
So the methylation that occurs in parent strand.
Yes.
In which part? Adenine. Adenine undergo methylation here.
Yes.
5' GATC. Yes. Then, so role of DAM methylase is methylation, and methylation is the purpose of methylation. Yes, purpose. What was the reason for this methylation? We have to differentiate parent strand and newly synthesized strand.
Yes, ma'am. Yes.
Okay. Then
Yes.
only we can find out the error. Error
Yes.
is present in which strand?
Newly synthesized.
Okay. Error is present in newly synthesized. You can see this. Error is present in newly synthesized strand. Yes.
Yes. Yes, ma'am.
Here,
Yes.
error is present, and we cut that region. Okay.
Yes, ma'am.
Cleavage of backbone in the vicinity of mismatch and removal of T by exonuclease. Okay. This
Yes.
here we can see guanine and thymine. Is it correct? It is
No.
mismatched.
Cytosine. Yes, ma'am. Mismatch.
Yes, here it is cytosine. We need
Yes.
cytosine here, not thymine.
Yes.
Clear?
Yes.
Yes, it is mismatched. Mismatched. Okay. So we have to remove this thymine. Clear?
Yes, ma'am.
Now it is clear. I think it is now it is clear.
Yes.
This is the region a loop formation occurs, and we remove that part contain thymine. Okay.
Yes.
Then the remaining part is synthesized by nucleotide is synthesized by DNA polymerase 3.
Yes, ma'am.
Okay. Okay. Then next, what is HNPCC?
Lynch syndrome.
Lynch syndrome. Okay, okay. It is a big name. So first one is H, always associated with hereditary in case of small bio. Okay.
Yes.
Hereditary. Hereditary.
Non-polyposis colon cancer.
Okay. This replication error are always enter into cancer. So hereditary non-polyposis colon cancer, HNPCC or Lynch syndrome. That is linked with faulty mismatch repair. Okay.
Yes.
One of the most common inherited cancer, HNPCC. Okay.
Yes.
Then what is the role of, so here also I write C, it is L. Role of mute S, mute L and mute H.
They recognize that mismatch and mismatch pair, and they cleave to the parent DNA.
Okay. They find out in this diagram, it is clear. In this diagram, it is more clear. You can see when this proteins, green, yellow, blue, white, these three protein, mute S, L and H, when it binds, a loop formation occurs. Yes?
Yes.
A
Yes.
loop formation occurs and strand removal occurs. Okay?
Yes.
That's why I draw two diagrams because then only you can understand this loop. Okay? When
Yes.
this protein binds, loop formation occurs and this loop is removed. Okay?
Yes.
That then corrected strand will be there after repair. Okay.
Yes.
Okay. I think it is clear. Mismatch repair is clear.
Yes.
Yes. Yes. Next, any doubt?
No.
You want to explain more? Anything about this mismatch repair? It is one of the most frequently asking question for short notes. Mismatch repair. The most important DNA repair mechanism is mismatch repair. It is very common. That's why mismatch repair is an important part. Okay.
Yes.
Short essay we can ask DNA repair.
Yes.
University question. DNA repair mechanism is a repeated question. DNA repair. But short notes, in case of short notes, the most important part is mismatch repair. Okay.
Okay. Yes.
Okay. Then next we are entering into base excision repair. Okay.
Yes.
It's another process. Base excision repair is another process, that is depurination of DNA. Depurination of DNA. It's a spontaneous process or by the action of deamination or alkylating compounds which occur at a rate of 10,000 per cell per day. So this is also common. 10,000 per cell per day. Depurination of DNA. What is meant by this depurination? Thus, cytosine, adenine and guanine bases spontaneously form uracil, hypoxanthine and xanthine.
Yes.
Clear, Sandhya?
Yes.
Which are the normal bases? We know adenine, thymine, cytosine and guanine. Yes. What happens in this repair is that what happens in this condition that is converted into another form like uracil. Uracil actually it is present in RNA. Yes?
Yes.
Yes. So if uracil is present, it is wrong in case of DNA. Then hypoxanthine and xanthine, all these are error. So this type of bases, some cases spontaneously forms, no need of enzyme, spontaneous formation. In some cases deamination, some cases alkylation. Okay.
Yes, ma'am.
Okay, then this abnormal bases, actually they are not normal bases of DNA, but we have to remove that basis. What happens in case of mismatch repair? Mismatching of base pair occurs, yes?
Yes, ma'am.
Adenine with the thymine instead of cytosine is there. Adenine with cytosine.
Yes,
Okay, otherwise cytosine with the thymine
Uh-huh.
instead of guanine. That is known as mismatch repair.
Yes.
What is the difference between mismatch repair and a base accession repair? Here what happens in case of base accession repair? What happens?
They spontaneously, the bases are form uracil, hypoxanthine and xanthine.
Abnormal bases are formed. That is more important. Okay,
Yes.
they are not normal bases; they are abnormal bases. Instead of cytosine, adenine, and guanine, that is converted to uracil, hypoxanthine, and xanthine. By which process? There are three mechanisms for this formation of abnormal bases. Which are the three mechanisms? Sandhya.
Hello, yes, ma'am.
Okay, which are the three mechanisms for the formation of this abnormal bases? That may be spontaneous process or
Yes,
deamination or
ma'am. Examination.
alkylation.
Okay,
Okay.
okay. Yes, ma'am.
Then one special type of enzyme is there that can remove these abnormal bases. In mismatch repair, we discuss three proteins. Yes,
Yes, ma'am.
mutase, mutel, mutH,
Yes, ma'am.
SLH. Okay, but here one enzyme is there. That particular enzyme is known as N-glycosylase. Okay, that is specific for each basis. Specific N-glycosylase can remove this abnormal basis. Okay,
Yes, ma'am.
specific N-glycosylase. Okay, then what happens? A defective DNA when for example, a defective DNA in which cytosine is deaminated to uracil is acted upon by the enzyme uracil DNA glycosylase. Result in the removal of defective base uracil. Okay.
Yes, ma'am.
Okay. This creates epurinic or epyrimidinic site. What by epurinic or epyrimidinic site?
That
Devoid of A or G. Yes.
Yes, ma'am.
Devoid of C or G.
Mm.
That commonly referred to as AP site. This terminology is very important. In case of molecular biology, we introduce some special terms. Yes.
Yes,
Like neat, ligation, like that. Okay.
yes, ma'am.
Okay. So here we introduce another term, AP site or a basic site.
Yes, ma'am.
What is it? That is epurinic or epyrimidinic site.
Yeah, it does not have adenine or guanine. Apuric.
Ah.
Apyrimidic does not have C or G.
Okay. Then what happens? It's a little more complicated. An endonucleon, not complicated, it's a simple process, but the steps are more. Okay. An endonuclease that cut the backbone of DNA strand near the defect and removes few bases. Okay.
Yes.
The gap so created is filled by the action of DNA polymerase and DNA ligase. Okay.
Yes.
So here we can explain it with diagram. Yes, this diagram is more easy than next the diagram. This is the normal DNA. Okay?
Yes.
Yes. Any problem in this case of normal DNA? Thymine, adenine, cytosine, guanine, thymine, adenine.
Normal DNA.
Normal DNA. But the C that is in blue color, because here the abnormal bases stands for abnormal bases. What happened to cytosine? Cytosine undergo deamination and it is converted to uracil.
Yes.
Then it is converted to defective DNA. It is normal or defective?
Defective.
Okay. So we have to remove this base. Yes, uracil.
Yes,
so yeah, what happened? Specific N-glycosylase. In case of uracil, it is uracil DNA glycosylase.
Yes.
Yes. So the uracil is removed.
Yes, ma'am.
So this site, one pyrimidinic base is absent.
Absent. Apuric site.
epipyrimidinic site.
Sorry,
Yes.
pyrimidinic.
It is not epipurine, it is epipyrimidine site.
Yes.
So that particular site is known as AP site
Yes, ma'am.
or a basic site. That
Yes.
base is absent. That's why a basic site.
Yes, ma'am.
Okay. So we have one enzyme. That enzyme is known as endonuclease. Okay.
Mm.
What is the role of endonuclease? When we study the action of endonuclease, cutting occurs. Okay.
Yes, ma'am.
Endonuclease cut this area and this area and this TT, both
Yes.
T, remove this portion. Okay. TC 0, there is no base. T is removed.
Yes, ma'am.
Okay. Then there is a gap. Here is a gap.
Yes, ma'am.
Yes.
Yes.
That gap is filled by DNA polymerase and DNA ligase for nick joining.
Yes, ma'am.
Now back to the normal DNA.
Yes, ma'am.
So I think base accession is clear.
Yes, ma'am.
Yes. The same explained here also. We can see guanine, uracil. What happens? Uracil is removed by the action of uracil DNA glycosylase. This is the like the DNA structure. This explained like DNA structure. So here guanine, there is no uracil.
Yes, sir.
Here, okay, there is AP endonuclease. We named this endonuclease into more specific AP. Okay.
No.
Okay, AP endonuclease. Then what happens? This area is removed. Cleavage of phosphodiester bond. Okay.
Yes.
In this structure, we are entering more into the structure. Yes.
Yes, ma'am.
So here cleavage of phosphodiester bond occurs. Then again the actual cytosine is added.
Yes, ma'am.
So I think base accession repair is clear now.
Yes.